be looking for information about what kinds of chemical bonds bond bases to bases. So we're talking about the structure of nucleic acids. One of the parts of the structure of nucleic acids are the bases. So what kinds of bonds bond a base to a base? And then what kind of bonds bond a phosphate to a sugar? Other things to be looking for: what component of a nucleic acid determines its name? Our nucleic acids are DNA and RNA. Their full names are deoxyribonucleic acid and ribonucleic acid. So why are they named differently? And what determines the directionality of a DNA strand? We're going to talk about a lot during the semester. There's 3' to 5' and 5' to 3'. So what makes a strand 3' to 5' or 5' to 3'?
Okay, so today we get to start digging into molecular genetics, which is honestly my favorite part of this course. We're going to step through talking about the components of DNA. And a lot of this is hopefully a review, but I'm going to emphasize some points that are really going to be critical for understanding DNA replication and then transcription and translation, and also things like crossing over and recombination and how those work. So often whenever you see DNA depicted, you're going to see it looking something like this. And what we're going to focus on a lot of the lecture today is this little piece of the DNA double helix. We're going to break down each and every one of these components because they're all going to be really important to the things that we're going to be talking about in the future. This little piece of DNA is called a nucleotide, and a nucleotide is the building block of DNA. So DNA is made up of a whole bunch of nucleotides. So this little part that I've outlined it here in red, we've zoomed way in on it here, and we're going to talk about what each of these three parts are and some important pieces of those three parts of a nucleotide. The first one we're going to talk about, I've outlined here in red. This is called a nitrogenous base, and I'm sure you've heard about these a lot. And the nitrogenous bases are adenine, thymine, guanine, and cytosine. You often see them represented by A, T, G, and C. So this part of the nucleotide is the nitrogenous base, and the nitrogenous base is what actually makes the code that gets read by the cell to produce proteins. So the order of the nitrogenous bases, and we've talked about this briefly before, is what makes the code. And we're going to talk about that in detail when we get to transcription and translation. So why the heck is this thing called a nitrogenous base? I just want to highlight that the nitrogenous bases are very, very nitrogen heavy. So at each of these corners of this molecular structure that this is representing, you have a nitrogen. So these are very nitrogen-rich structures. There are two different kinds of nitrogenous bases. You have nitrogenous bases that are made only of a single ring. This is cytosine and thymine are single ring nitrogenous bases. And these single ring nitrogenous bases have a special fancy name. They are called pyrimidines. So single ring is a pyrimidine. And this is cytosine and thymine. And you'll notice here it says DNA only. So when we get to RNA later, we're going to add another pyrimidine. So there's a different pyrimidine in RNA. So just kind of keep that at the back of your mind. The other type are the double ring, adenine and guanine. These have two rings, and these are purines. And the way I remember this is basically the size is backwards. The actual size of the base is backwards to the size of the name. So purine is a small word, whereas the purine bases are larger. They have two rings. Pyrimidine is a big word, whereas the bases are small. They have only one ring. If you have a better way, awesome. I've my entire career, I've tried to come up with a way, like a little something to help remember that, and that's the only thing that I've come up with. Okay, so purines and pyrimidines. So purines and pyrimides have complementarity. So there's these bases only bond to specific other bases. And the base that they bond with is complementary. And so what that means is that pyrimidines bind with purines and purines bind with pyrimidines. And a specific purine binds with a specific pyrimidine. So in this case over here, guanine is always going to base pair with cytosine, always. G will always base pair with C. It's complementary. So we've got a purine, see there's two rings here, bound to a pyrimidine, which is a single ring. And this guanine purine will always bind with cytosine. Over here, we have adenine bound to thymine. And so we have a purine, adenine, bound to a pyrimidine, thymine. And that is the way they will always base pair. And that's going to be important later when we start talking about DNA replication. Also, right through here, where you see our purine, guanine bound to our pyrimidine, cytosine, these little dots, what they represent is a hydrogen bond. And so across DNA, the things that kind of make up what we often refer to as the rungs of the ladder, those bonds are hydrogen bonds. And hydrogen bonds in the world of chemistry, I'm sure you've learned this, are pretty weak. But collectively they provide stability for the DNA. But the fact that these bases are bound by a pretty weak bond is actually what allows DNA to expose these bases and allow the cell to read the sequence of bases. And again, we'll come back and we'll talk about that in more detail. But keep in mind that these bases are bound together by hydrogen bonds. That will definitely come up on quizzes and homeworks. And just to remind you once again, another visual, which are our purines and which are our pyrimidines, showing you them again in kind of a different color and a different chemical structure representation. So those are the bases, the nucleotide, the nitrogenous bases, lots of nitrogens. We have purines and we have pyrimidines held together across the DNA double helix ladder with hydrogen bonds. The next important part of a nucleotide is this sugar. So a nucleotide is always going to have a sugar, and it's always going to have a pentose sugar. In DNA, this pentose sugar is called deoxyribonucleic acid, and that's where DNA gets its name. So it gets its name from its sugar, deoxyribonucleic sugar. So this pentose sugar, pentose means that it has 5 points. So 1, 2, 3, 4, 5 right here. It's a pentose sugar. And another thing is very often when you see any structure, but especially a sugar like this, you're just going to see like corners. You're just going to see it represented this way, and there's no label at these corners. Well, what's actually at these corners is carbons. Carbons at each corner. And these carbons are really important. And these are really important when we start talking about the directionality of DNA. So I'm going to go through this, and we're going to come back to this over and over and over again. So these carbons are numbered. And we start at the point, the oxygen is at the point of the pentose sugar. And we go clockwise, or we go to the right. So we start with the oxygen, and then we go to this first carbon. It's number 1, so it's the 1' carbon. And then we come down to the next one. We're moving clockwise, the 2', the next is the 3', the next is the 4', and then we have another carbon kind of sticking up off here like a flag. This is our 5' carbon. And this may seem like, why do I even care, or I need to know this? This is chemistry. Yes, and this chemistry is critically important to the structure of DNA. So make sure that you're comfortable with this concept of having these carbons that are labeled in this order, because we're going to come back to this over and over and over again. And understanding the way the DNA replicates itself, you have to make sure you're comfortable with this. And I'm going to explain how and why. Okay, so that's our pentose sugar. And then last but not least, a phosphate group. So the three parts of a nucleotide are the nitrogenous base, a pentose sugar, and in DNA, this pentose sugar is dextrose. I think I just named DNA earlier, didn't I? I said deoxyribonucleic acid. That's the name of all of DNA. The sugar is dextrose, so that's where the deoxy comes from. I apologize. Okay, here, phosphate group is the last component of our nucleotide. All right, oh, sorry, that's not supposed to be there. I'll fix that. Going back to looking at the whole DNA molecule, and this is very often how we see it depicted, and we'll a lot of times see it just sort of wound up like this. And what this image is showing you is if we look at the actual molecular structure, what we're seeing is nucleotide bound to 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 nucleotide. And these make up the structure of the DNA. This part right here is called the backbone. And the backbone of DNA is made up of the sugar and the phosphate. So it is often called the sugar-phosphate backbone. So this part is the backbone. So in this little part right here, the bottom part, that little sort of ribbon-looking thing, that's the backbone. And that's where the sugars and the phosphates are. And then if you look right here, we often hear these called like the rungs of the DNA, as if they're the rungs of a ladder. So this is where our bases actually are. So here we have G, a guanine, bound to a cytosine. And then here we have T, a thymine, bound to an adenine. And these are bound across the two backbones. So if we look up here at this with the components of the nucleotide exposed, here this pentose sugar, 5-sided sugar, and here's your phosphate, so that's the backbone part. And then over here is adenine, your base, bound to thymine with hydrogen bonds across the middle of the DNA. And then over here, we're back into the backbone. So here's the pentose sugar, and here's the phosphate. That's the part that makes up the backbone. So the nucleotides themselves, right here, are connected to each other. So here's the sugar phosphate and its base. So this nucleotide is connected to this next nucleotide by phosphodiester bonds. And these phosphodiester bonds are very strong bonds. So this is very different than these weak hydrogen bonds across the middle of the DNA. These phosphodiester bonds are very strong, and you can kind of see that it's represented by solid lines here, rather than these dotted lines that represent the bonds across the middle of the DNA molecule. So to show you that, depicted a little bit differently, so what we're looking at here is the exact same thing we've been talking about. Here's our base, so this would be thymine. Here's our pentose sugar with all of our carbons. 2 of them are labeled. The 1' carbon is labeled and the 3' carbon is labeled here. And then, so this base and this sugar belong to this phosphate. And then this bond here, right here, to this next phosphate bonds this whole nucleotide to this whole next nucleotide. And so this bond right here is a phosphodiester bond. It's a very, very strong bond. So this nucleotide is bound to this next nucleotide through a strong bond. Okay, another thing about the structure of DNA that's really important, and you'll see this a lot and you've probably heard this this talked about before. It took me years to understand what the heck people were talking about when they said 5 prime to 3 prime or 3 prime to 5 prime. What the heck does that even mean? So if we look at DNA, so this is a double helix. There will be one strand that's running what we call 5 prime to 3 prime, and there will be another strand that's running what we call 3 prime to 5 prime. And the way we get that those names, the way we get that directionality is based on the position of this pentose sugar. So if you remember, I told you that those carbons are named. So let's zoom in and look at this. So remember that we start with the oxygen and we number our carbons. 1, 2, 3, 4, 5, and they're all prime. And so if we look at this strand of DNA, we can do that. We can number our carbons on this pentose sugar. So our oxygen is here at the kind of like little top, the peak of this pentose sugar, and we go 1, 2, 3, 4, 5. So the carbon that's at the top of this sugar is the 5 prime carbon. So we go 5 prime to 3 prime. And that's going to be the same here too. This one's going to be 5 prime and this one's going to be 3 prime. Same here, 5 prime, 3 prime, 5 prime, 3 prime. So then this strand is described as running 5 prime to 3 prime because of the direction of the carbons in the pentose sugar. I'm going to show you that again with a slightly different image. So here's the 5 prime end, and that's because right here is our 5 prime carbon, and then here is our 3 prime carbon. And then same thing here, here is our 5 prime carbon, and here is our 3 prime carbon. So this is running 5 prime to 3 prime. That's what 5 prime to 3 prime means. So it's named for the carbons on the pentose sugars. So once again, showing you that kind of all together, 5 prime, this is the 5 prime to 3 prime strand. And then the other strand, it kind of looks like those sugars have been flipped upside down. So here, if we, I don't know if you can hear my dogs barking, but they're losing it outside. Someone dared to let their dogs out. So here is our pentose sugar. And remember, this little peak is where our oxygen is. And so if we look over here on the other side, our little peak is flipped upside down. And so here, our 5 prime in this particular visual is at the top. Of course, we could turn it sideways, whatever. It would still be the 5 prime to 3 prime strand because the 5 prime would still come first. Now, if we flip our sugar upside down, our pentose sugar upside down, then our 5 prime is instead here. It comes second, you could think of it as we're reading this strand, whereas the 3 prime sugar comes first. So this strand then is 3 prime to 5 prime. We say it runs 3 prime to 5 prime, whereas this strand, because the 5 prime comes first, it's at this little bend in the elbow. It runs 5 prime to 3 prime. I'm emphasizing that because that's really important. So what we say about DNA, because of this 5 prime to 3 prime on one side, the other side is going to be 3 prime to 5 prime, is we say that the backbones of the DNA run anti-parallel. And so a way to think about this is that they run head to butt. So the head of one strand is next to the butt of the other strand, and the head of that strand is next to the butt of the other strand, like these two zebras. So this is the anti-parallelity of DNA. And in the next lecture, just in a moment, when we talk about DNA replication, this is going to be really, really important.